It's my pleasure to introduce as our next speaker, Shannon Corrigan from the Minderoo Foundation. She comes to us from very far away, from Perth, Australia, and we've worked with Shannon and her group for a few years now, and are really excited that you were able to come to PAG. I understand it's your first time at this conference, and looking forward to hear about the Oceanomics initiative. Shannon.
Okay, thanks very much for the introduction, Jonas, and thanks everyone for coming along. So as Jonas said, my name's Shannon. I'm a research manager at the Minderoo Foundation, and I'm also the Oceanomics Centre lead at the University of Western Australia. I do come from Perth. I live in Perth. I lived in the US for a while, but I never quite made it to PAG while I was here. And don't worry, I have plenty of maps in the talk so you can orient yourself to where we're coming from and what we're talking about. So today what I want to do is just give you a little bit of an overview of the work that we've been doing to generate high quality reference genome resources for marine vertebrates, and then I'll also put this in the broader context of the work that we do in the Oceanomics program around eDNA. So I'm going to start off with some acknowledgements because they're super important, and I'll run out of time if I don't do them front-loaded. And then I'm going to give you a really whirlwind overview of the reference genome program, and then some progress that we've made since we began sequencing towards the end of 2022. It will be whirlwind, so if I touch on anything only briefly, just due to time constraints that really piques your interest, I'm around for the rest of the week, as is my colleague Leanne Ernstus. Please do come and chat to us. We want to hear about what you're doing and share our experiences while we're here at the meeting. And then finally, I want to get to the more interesting part, which is telling you some of the stories and takeaways that are starting to emerge from the data that we've been collecting for the past couple of years. So I first want to begin by acknowledging the Wajari, Menang and Wajak Noongar people, the Coco Malay people, and the local communities on both Cocos Keeling and Christmas Island as the traditional custodians of the lands and waters of the places that we live and worked in order to generate the data that will be forming the basis of my presentation today. I pay my respects to their ancestors and elders and extend that same respect to any First Nations peoples that are present here at the meeting. I also want to recognise all of my lovely colleagues here, either named or pictured, as this is they've really contributed most of the work that I'm going to be presenting. It's my privilege. We've all been on a very steep learning curve over the last couple of years, so I'm really proud to be able to stand up here and present on their behalf. And then I also need to acknowledge, of course, everyone at PacBio and Millennium Science, who they partner with down in Australia, and also EBP communities, because we've become an affiliated project and have been really fortunate to be able to walk in the footsteps of colleagues at both the VGP and the Tree of Life, and that's allowed us to really hit the ground running with a lot of the things that we're going to be presenting today, and so I'm very grateful. Okay, who is Minderoo? How do they work with UWA, and what the heck is Oceanomics? So Minderoo Foundation is a modern philanthropy that's based in Perth, Western Australia. It organises itself around 3 focal areas, aiming to uplift communities, support gender equality, and protect natural ecosystems. They also aim to respond to emerging threats. Oceanomics is one program that sits under that protecting natural ecosystems focus area, and it has a broad aim to really develop environmental DNA biomonitoring as a scalable biodiversity sampling solution. And there are a number of aspects to the program, which goes from everything to do with field sampling. I would have pointed, if I could have pointed, to this infographic, but we do field sampling to bringing marine eDNA samples and faunal samples into the lab and doing genomic sequencing, computational data analysis, translation of that data into ways that it can inform decision-making and policy frameworks in the marine conservation space. The middle piece of that is all around data generation, and that happens in partnership with the University of Western Australia via the Oceanomics Centre, which is our onshore lab-based facilities based there at the university. So the key activities that happen at the Oceanomics Centre that's where the powerhouse where we generate our reference genome data and also produce marine biodiversity eDNA data sets at scale. So that brings me finally to ocean genomes. So when we first started working in the eDNA space, we, like everyone, really quickly realized that existing reference databases are really insufficient for us to comprehensively identify the unknown fragments that we're retrieving from eDNA samples. We were particularly interested in marine vertebrates, And at the time when we started working on this, more than 96% of those lacked a whole genome reference sequence. And we also noticed there was a sampling bias in available whole genome reference availability, in that most of the available sequences came from really well-resourced regions. They usually represented pretty common species. And of particular interest to us was a big black hole sitting around the Indian Ocean, which was particularly relevant because of our geographical proximity to that area, but also because we know it houses a lot of unique fauna. And so the Ocean Genomes program was spun up really in order to address that void. And it's really grown over the last couple of years to become a major feature of the overall oceanomics program. So the main aim of this part of the program is to accelerate and scale the production of high-quality reference genome resources for marine vertebrates. Given the initiation story for the oceanomics program, this was mainly to facilitate breakthrough eDNA monitoring applications, but of course we recognise that these types of reference materials are really much more useful as a foundational scientific resource that can inform advanced science, but also inform conservation and management of these organisms. So we've adopted a tiered approach to reference resource generation in ocean genomes. So the primary goal is to generate high quality, near end-to-end reference genomes for a representative species of all marine vertebrate families. And then we want to make sure that we're dedicating those resources in the areas that they're really needed most. So we put special emphasis and priority on threatened species, but also anything that's high conservation value. And so for this part of the program, we've aligned with the EBP and becoming an EBP-affiliated project, and we're aligning with all of the methods and quality metrics and standards that are proposed by the VGP to ensure that when we are investing in a reference, that it can really give it the best shot at being the best quality that is possible. At the other end of the spectrum, though, we realise that sample quality can be prohibited to generating some of genomes using HiFi sequencing, and so we're doing a draft genome assembly from some of those samples that is of lesser quality really offers us a way to represent a more comprehensive sampling of species diversity in our reference databases. So we will do that sometimes in situations where we can't go for a high-quality genome. So with this kind of strategy in mind, we've developed some pretty key capabilities in the reference sequencing space at the centre, two major workflows that are largely overlapping. So we're even embedding best practices for sample acquisition and sample preservation and extraction of high molecular weight DNA in the lab. On the high-quality reference genome side of things, our standard approach is to combine PacBio HiFi sequencing with Hi-C proximity ligation data generated via usually Dovetail, OmniC, or now their link protocols. On the draft genome sequencing side of things, we usually just opt for 50x and Loom and a short-read data. We've developed some custom pipelines for reference genome assembly and QC, curation remains largely manual in our hands, and then we're making that data available via a number of different custom and public repositories. So we've made a few key improvements over the last several months in order to move from a kind of onboarding the technology phase and into a scaling phase for data production. The first is about a year ago, I think we upgraded to the PacBio Revio system. We're a relatively small team, and so although we have a lot more capacity on that instrument, it's so far we're seeing increases in our throughput of about 5-fold as a consequence of doing that. Of course, I'm now on the search for finding more and more samples to continue feeding the beast. And so adding ultra-low input protocols to our workflows has really been enabling because now, up until sort of this period, we've been parking those really challenging samples to the side because they're kind of resource limited. And so now it's really starting to unlock access to a wide range of more diverse sample types in our hands. And we're starting to see some nice sequences emerging even from our most precious specimens. Another thing we've been working on is automating some of our library preparations on our Biomac i7 liquid handler, and you can see in the table there a list of some of the library preps that we've automated, which includes an end-to-end walk-away process for PacBio SmartBio library preparations. And so we can now handle up to 96 samples Not that we're usually ever in a position to have that many on the go at one time, but it's possible. But the key here is that it's reducing the processing time and then allowing that to be walk away so that our staff can go and focus on other things. And then the other thing that we're doing that's really enabling is we've put together some scalable and reproducible pipelines for reference genome assembly, draft genome assembly, and also minor genome assembly. And we're either have or are in the process of transitioning those pipelines into Nextflow and then making them available via NF Core. So if anybody wants to check those out, they're for now available on our GitHub. Okay, so what have we actually done besides all of those things? So what you can see here is a graph that's showing our progress on both the high-quality reference genome generation and draft genome generation over the last 2 years. So it's like Global Genome Sequencing Consortium, we kind of track our progress at the family level for high-quality genomes, species level for draft genomes. So you can see here in grey are all of the about 495 marine vertebrate families that don't have an available reference sequence. In purple are the proportion that has a reference available. And then in blue, you can see the proportion that's being worked on currently by Ocean Genomes. This doesn't necessarily mean complete, but it means somewhere in our workflow. Some of them are complete. But to date, we've generated high-quality reference genomes for about 56 different marine vertebrate families. I mentioned that we put emphasis on those things that are of high conservation value, regional endemics, things like that. So, of course, like everyone, we've done more than just the family representatives. We currently have somewhere in the works high-quality genomes for 104 different species. So in the in the draft genome space, obviously there's a lot more marine vertebrate species, about 40-something thousand. Actually, no, 20,000. Sorry, I added in freshwater fish there in my head. 20,000 marine vertebrate species. And so our contributions in this space so far have been sequencing 358 different species at the draft genome level. And we have multiple representatives of some of those, so 699 draft genomes. So this slide is really just to give you an idea of the kind of diversity that we're capturing in our sequencing efforts, and to just show with a pretty figure that we are really hitting those goals of kind of emphasizing organisms that are of high conservation value and those first representatives of a family. So our data today includes a range of different food fish species, which Jonas mentioned at the beginning of the talk. Sorry, now you're going to have to know your fish because I can't point to which ones those are. But also some wide-ranging pelagic species, some elasmobranchs, so cartilaginous fishes, sharks and rays who have nice big squirrely genomes, and reef-associated species. So what do they actually look like? So this is the first 3 that we did in the lab, and we think it's going pretty well. So these are genome quality metrics for these 3 species. Here we have an old wife, which is a monotypic family, so it's the only representative of that family as a species. It's really commonly found in southern Australia. A rough bullseye, which is a southern Australian regional endemic. And then on the right-hand side is the spangled emperor, which is a very common commercial fish in Western Australia and also the Indo-Pacific. So these look really very nice with our approach of using HiFi and Hi-C sequencing. But we're kind of bracing ourselves and starting to see, like, as we move into the more challenging genomes, especially elasmobranchs that have larger genomes, that obviously we're facing all of the same challenges. So I'm very keen to chat to people and hear about their experiences so we can learn from you all while we're here. Okay, so where can you find all of this data? So we have a commitment to open science and equitable access to data and benefit sharing, and this is really the next big milestone for us. So we've been kind of drinking from the fire hydrant for 2 years, getting everything up and going, and now it's time to do a big genome dump. So that's coming in the next weeks to months, and then we'll be available via these channels here, NCBI, at the Bio project there. You can also track our progress at Genomes on a Tree. And then we also have a custom dashboard, which includes some eDNA data as well as our reference genome progress. I wish I had time to show it to you. It's really cool. You can go on and explore environmental DNA data that we've sampled, and you can explore our genome data. And we have a little custom AI chatbot there, so you can actually ask questions of the data and explore it that way. So if you get a moment, it's in a beta release. It'll get better over time. So give us that grace, but feel free to find that online and have a look. Okay, so that's what we've been up to. So now I kind of want to pivot and talk more in the context of the broader oceanomics program. So over the last couple of years, the broader program has been partnering with the Australian government, Parks Australia, to conduct some marine eDNA biodiversity campaigns at a range of locations that you can see here on the map throughout the Australian Marine Park Network. And what I want to focus on now is just two of these campaigns. One's the Indian Ocean territories, which you can see out there in the northeastern Indian Ocean, and the Great Southern Reef in the southwest of Australia. And the reason I want to focus on these is because it's really nice examples where we've been able to combine environmental DNA sampling together with tissue and specimen collection for high-quality reference genome generation. So the Indian Ocean territories includes Cocos Keeling Island and Christmas Island. They're about 3,000 kilometres off the coast of Western Australia in the northeastern Indian Ocean. So they're a ways out. They're actually closer to Indonesia than they are to Australia, but they're Australian territories. So Cocos Keeling consists of 2 atolls and 27 different islands. It's characterised by shallow water, sandy beaches, reefs and seagrass beds. Christmas Island, on the other hand, is quite different. It rises really sharply as a single island mass. In the middle of the Indian Ocean, it's surrounded by very deep tropical water. So even though they're geographically proximate to each other, they represent very different environments, different histories, different ecosystems, and different faunal assemblages. They also include different cultural and historical significances. And so both of the communities on each of those islands maintain really strong connections, both to the islands and also to the marine fauna surrounding those islands. And for that reason, the Australian government has slowly been transitioning management of those regions back into the local communities in the Indian Ocean territories. And so both of those communities are interested in using environmental DNA to survey their regions, and so we were able to partner with them to go and survey both of the island groups, and we collected 345 eDNA samples and 232 tissue samples in collaboration with those to characterise the marine vertebrate assemblages around the islands. So this is some kind of brute force results. So on the eDNA side, we were able to sample 65 sites, 345 samples, as I mentioned, and make detections from that data for 125 different marine vertebrate families, representing 454 species. On the genome side of things, we were able to collect tissue samples from 54 different species, and then the quality of those allowed us to do reference genome sequencing at high quality for 27. 9 of those were first-in-family representatives, and we also produced 21 draft genomes from some of those samples. So this horrible Venn diagram in the middle of the slide is basically just showing you the different assemblages across the island. So when we do eDNA, we use 2 metabarcoding markers targeting 2 mitochondrial regions, 12 and 16S. So on that side, the right-hand, your left-hand side is Cocos Keeling Islands, on the left is Christmas Island. And you can start to see that in our eDNA and the genomes that we sampled, it's reflective of those different environments and assemblages. So from Cocos, we see more reef-associated species, inshore species, including this humphead Maori wrasse, which is culturally important to the people there, but also quite endangered. Whereas in Christmas Island, we see detections of much more very pelagic species, wahoo, tuna, all the way down to deep water. These are all deep water snappers that you can see in the middle there. And then this weird little beardfish at the bottom. So I wanted to talk a little bit more about the beardfish because it ended up being quite an unusual finding when we were there. So this is a species of polymyxia, and originally we nominally identified it as Polymyxia burndi, given that that species has an Indian Ocean Indo-Pacific distribution. It's a benthopelagic deep water species, and we ended up sequencing this at high quality because it was going to be the first representative of both the order Polymyxiformes and also Polymyxidae. And so we generated a nice genome from this guy, and so immediately, because it was going to become an authoritative reference, I wanted to confirm that identity. We weren't able to keep the specimen as a voucher because the local fisher donated it and he wanted to retain the specimen. And so we pulled out some of the barcoding genes and did a validation against available data to check whether our ID was correct on this specimen. And so what you can see here is a tree from all CO1 data for all of Polymyxia, the genus, that was available on Bold. If you have a super good eye, you can see that this group is subject to a lot of taxonomic confusion and cryptic species, and that's reflected in some of the names on the branches. But what you can see on this side of the tree is the updated namings based on a recent taxonomic revision. Down the bottom in green is that burn die group where we expected that our specimen would fall, and highlighted in blue is where our specimen indeed fell. And so this was really surprising because that group is Polymyxia holisterae. It's a very recently described species. It's only known from 3 specimens, and they were all recorded in Bermuda and the Gulf of Mexico. And we're out here picking up this fish in the middle of the Indian Ocean. So unfortunately, we didn't have a specimen, but we can zoom in on the image of this guy and see that indeed it has a very large eye, and that the dorsal soft ray counts are consistent with what was seen in those type specimens for were collected from Bermuda. So why do you care? I think there's about 5 people, including me, that care about beardfish. But if this is indeed this lineage, it represents a tens of thousand kilometre range extension for this species. And it would also imply that even in deep water species, potentially this area around the Indian Ocean and Christmas Island is representing either a hybrid zone or a region of mixing, as it is for the more shallow water reef species. And so we wanted to look at our eDNA data and see if we got any other detections. So we detected this from the specimen, so we wanted to look at the eDNA and see if we found it anywhere else. And so what you can see here, yes, we detected 4 different sequence variants in our eDNA that mapped to polymyxia. And then the size of the bubbles relative to how many detections we had of it and the locations they were sampled. So all sampled around Christmas Island and in between Christmas and Cocos, but at multiple sites. And so on the bottom, these 2 sequence variants are mapping to this Polymyxia holisterae. So we've got multiple detections now of this weird Bermuda-type lineage popping up in the Indian Ocean. And then the 2 sequence variants on the top have a divergent identity. And so that's suggesting that perhaps we at least have multiple lineages occurring in the region, but also multiple detections of this weird thing that's shown up from Bermuda. So I just think this is a really nice way of demonstrating, even though it is just a beardfish that you probably haven't heard of before, but demonstrating how when you go to these really remote regions and you can combine specimen sampling with reference genome generation and biodiversity surveys, that we can really start to see the value of doing all of that in conjunction to understand diversity and distributions of marine vertebrate fauna. How am I going for time, Eunice? Because the timer hasn't started and I feel like... Okay, so I'm going to move through this super rapidly. So another place that we have done some sampling is the west coast of WA, the Great Southern Reef. So this is another marine eDNA survey that we were able to do, but we also collaborated with the Western Australian Museum. We're able to work with collection scientists down there that were doing in-water faunal surveys, super sharky down there, and so they were diving from shark cages and collecting invertebrates and fish fauna all the way along the southern coastline. So we were able to sample about 1,000 kilometres of that coastline. This is them jumping into the shark cage and dropping down to collect specimens. So we collected 845 eDNA samples around this range, and again, 162 tissue samples. And this was super, super valuable because this region is characterized by really high endemism. It's very rugged, it's hard to access. There's hundreds of uncharted islands and rocky outcrops, and so it hasn't been surveyed all that much. But it's a very biologically important area for things like white sharks, whales, and a lot of small endemic species. And so on the eDNA side, we were able to make detections from 144 different families, 349 species, and then we're able to generate high-quality reference genomes for 29 endemics and 20 first-in family. And again, all of these guys were representing these really southern temperate fauna and a lot of the regional endemics that were known to occur there, but that had been little seen before. So we did a similar thing for this Pemferis. We wanted to have a look at our eDNA and see more broadly where it's distributed, where we're seeing this pop up. So this was the first genetic data that was being added to public reference databases for Pemferis klunzingeri, so other species were available. And so in the top of this figure, you can now see that by adding that, we're able to make detections of that species in our eDNA data throughout the known range. And then if I had time to spend on this, I would tell you that we can also start to tease apart some of this data and use it as a way to triage which parts of the tree we might want to add references too. So we're seeing things matching to this Pemphiris schwenki down in the bottom right corner, but at low identity. And so you can start to use this kind of thing to like triage where you need to go to fill out the tree. Likewise, Pemphiris japonica in the middle there, we have a range of different identities, and that's indicating that perhaps some of the references have IDs on them that are incorrect. Okay, so I want to jump to this final slide because this is kind of the take home here. So one of the comments that we get a lot is, okay, great, but you don't really need high quality reference genomes to do eDNA, and it's a really fair point. You maybe don't necessarily, but we wanted to start thinking about in future-proofing two things: biodiversity surveys, biodiversity collections for long-read sequencing now, but also emerging technologies into the future, and also our eDNA surveys. You know, at the moment, we're targeting short regions in a metagenomic space, but that's not always going to be the case. We're going to eventually transition to wanting to use all of that material that's floating around in our marine eDNA samples. These are horrible figures. I'm going to do it very quick. We wanted to shotgun sequence some of our eDNA samples from both Indian Ocean territories and Southwest WA, so that we were capturing all of the DNA that was there. And then we created a metagenome of all of the available fish genomes on NCBI and added all of the fish genomes that we had generated in-house to that. And then we used a tool called SourMash, which basically compares metagenomes and genomes in order to do matching via k-mers. And so we just wanted to say, like, of all of our eDNA k-mers from that we can identify as being marine vertebrates from a shotgun sequence, how often are they being assigned to references that were already available versus how much more are we getting by adding these new references to databases. And what you can see, the columns on the right are basically the small ones blown up. And then I coloured the references for the fraction that came from either the Southwest or the Indian Ocean territories. And so it's looking really nice. So we only get a tiny fraction of our eDNA samples is marine vertebrates. That's not new. We know that it happens every time we look at a sample. However, it's not nothing, and there's a lot of reads there. And most often by adding references, the high quality from the regions where we sampled, we're then able to assign a lot more of those reads. And so there's a lot of value here. As we start to think about where we've come from and where we're moving to. And so we think about capacity building a lot. In order to continue this work, we're looking at putting modularized container lab spaces in remote locations in partnership with Parks Australia, but we're not the only people that are thinking about this kind of thing. And so I just want to end on a kind of call to action to thinking about things like Vega and how we can get this technology into these locations in order to, really move forward to a place where we're doing specimen sample collection and sequencing in place of provenance and future-proofing our biodiversity collections. Thank you. Sorry for going over time.